Yeah. Now, within 30 seconds, YouTube will start.
All right.
And then Dr. Amarja, she is going to start today.
Amarja Bhosle.
Amarja Bose.
Yes.
You can start, Amarja.
Yes, sir, yes, sir, sure. Okay. So very good morning, everyone. I'm Dr. Amarja Bhosle, Assistant Professor, Department of Microbiology from Vidya Pratishthan's Arts, Science and Commerce College, Baramati. Today I'm here to anchor this session because Dr. Jay is not able to join the meeting owing to certain circumstances. So today we all are virtually gathered here to attend a guest lecture by our eminent speaker, Dr. Noah Lal. She is working as associate professor in Department of Microbiology at AJ College of Science and Technology, Thokankal, Trivandrum, and handling both the UG and PG department very efficiently since 2005 till date. She dedicated 17 years in the teaching field, and she has pursued her PhD in zoology from the Thuronic VK Government Arts College, Trivandrum. So, in 2020, she achieved several achievements with special highlighting of the Best Program Officer National Service Scheme, University of Kerala. Dr. Noah created total 6 extraordinary records, especially as advisory committee member for the Guinness World Record official attempt, that is thickest book in the world, organized by the ESN publications. She has been awarded with 7 awards, and especially I want to mention here that the Women Research Award by the International Scientist Award on Engineering, Science and Medicine in the July 2021. She is recognized as editorial board member in various national and international journals. Dr. Noah dedicatedly worked on the 3 projects in the high quality projects. Of course, she has chaired various sessions in the several national and international conferences. She has published a total 29 publications in various national and international conference. She has published total 10 papers in conference proceedings, total 18 chapters in book, and then she has attended total 10 workshops. So with this very brief introduction, now today she is here to deliver her lecture on the topic anti-quorum sensing potential of the chosen marine sponges. So I would like to request Dr. Noah to start with her.
Thank you, Amarja ma'am, and thank you, Arvind Deshmukh sir, for inviting me to this lecture. I'm so happy to present in front of such eminent personalities. I don't know how better I'll be able to present, but I'll try my best. Since I'm having a sore throat and all, I'll try my best for the best presentation experience. Hope my slides are visible.
Yes, visible.
Yeah, okay, sir. First, I'll go into the topic anti-quorum sensing potential of chosen marine sponges. The reason behind why I went into this topic was since now antibiotic resistance is very common throughout the world, antibiotic resistance, and the property of antibiotic is to kill the bacteria, and as such what happens is in severe cases like septicemia and bacteremia, all this antibiotic killed organisms or their toxins, it will exist in our body and which will cause shock and all. From that concept, we came to a new technique, anti-quorum sensing. That means to break the quorum sensing. I'll get into the details. First, on what is quorum sensing? It is the cell-cell communication between all the bacteria. And for this quorum sensing, the specialty is that during quorum sensing, bacteria, they can mate each other, they can release toxins, they can cause diseases, that is their virulence capacity is enhanced, bioluminescence, then swarming ability, we can see that. And coming to the next slide, here we can see that for quorum sensing to occur, there should be a minimum number of bacteria. That is, when the bacterial count is very low in a particular place, quorum sensing can't occur. So the bacteria, their cell density should be more. That when a threshold concentration is reached, this bacteria, they communicate with each other by means of quorum sensing using autoinducers. Autoinducers are compounds like pheromones, like in animals, like pheromones. Autoinducers are compounds which can help the bacteria in their communication. But only when there is a threshold concentration is higher, this autoinducers are produced. The bacteria come into contact with each other and they start their mechanism of quorum sensing. And next we can see inhibition of quorum sensing. How can we inhibit quorum sensing? Since quorum sensing is a potent method for bacterial virulence inhibition, what we have to do is we have to find out quorum sensing inhibitors, which break the, they does not kill the bacteria. What they actually does is they just break down this communication. And now it's a trend. Now almost all are going into natural extracts. So from that, it's seen that this natural extracts, they can break the quorum sensing molecules. Means they can break. There are various activities in which they do it. Here are the strategies. All this are just a small introduction onto quorum sensing and quorum sensing inhibition. Strategies for quorum sensing inhibition. Actually, 3 strategies can be applied for quorum sensing inhibition. First is targeting the signal generation. For that, only if signal precursor is formed, signal will be eliminated, and signal will be delivered, and this signal binds with the signal receptor and quorum sensing occurs. So here we can prevent signal precursor from emitting signals. The next one will be targeting AHL signal dissemination. Here we can prevent signal from attaching to the signal receptors. And the third technique is preventing the signal from binding to the signal receptor. All these strategies are quorum sensing inhibition techniques. Now I will go into my topic, my PhD topic. The objective of my topic was to isolate, characterize novel quorum blocking molecules from marine sponges. Marines, the reason for selecting marine sponges is since there are a lot of new products from marine sponges, and it's the less explored things. And moreover, marine sponges, no need of going and collecting and all those things. It usually, when we go for fishing and all, some of these sponges, they attach the fishing nets and they come, they come to the shore. We can collect these sponges, select them, and isolate their compounds and do, make this quorum sensing inhibitors. Then second objective was to evaluate the quorum blocking potential of the marine sponge. I have selected here 5 sponges. I'll get into detail of all those things. From this 5 sponges, I have identified 2 of them are efficient, and in that one is more efficient. Then to screen and characterize the quorum blocking compound through spectroscopy studies. This was the objective of my PhD work. And these are the materials and methods I have used. First one was collection of pathogenic bacteria. Here I collected the pathogenic bacteria from shrimp. And my area of interest was Vibrio species. 5 Vibrio species. I have collected 5 Vibrio species. Vibrio cholerae, Vibrio algirolyticus, Vibrio parahemolyticus, Vibrio fishery. Vibrio fishery was kept as a control organism to study the growth of other organisms, other Vibrio species. Then the growth pattern. I have just checked the growth pattern of all these Vibrio species. And after that, I have characterized the isolated bacteria. I have to check whether the isolated bacteria is Vibrio. For that, I have done the biochemical characterization, then their antibiotic susceptibility test, then their molecular characterization by DNA sequencing and sequence analysis, construction of the phylogenetic tree, and later I went into the characterization of acyl homoserine lactones. This acyl homoserine lactones, or AHL, these are the communication molecules or the autoinducers, which I have previously mentioned. These autoinducers are responsible for the communication of bacteria. So I had to characterize the acyl homoserine lactones by TLC, HPLC, and GCMS. Then my second topic was, second area was screening of biologically active anti-quorum sensing agents. For that, I had to collect sponges. All those was eco-friendly collection based on the season and location. Eco-friendly collection is that during fishing, all these sponges, since they are sessile, they attach to these fishing nets, and we can collect it from that. It was collected according to season and location. Sponges were identified. These sponges were extracted using methanol. Methanol was the compound. I have used different compounds, but methanol was found to be more effective. Then I have done the bioactivity screening. Bioactivity screening. That is in vitro antibacterial screening by agar punch hole method. Then I have determined the MIC, minimum inhibitory concentration of this sponge extract. Then after that, I have gone to the administration of sponge extracts with different Vibrios to study their characteristic. Then I have done the AHL degradation assay. For that, I have used micro titer plate assay, chemiluminescence assay, reporter bioassay, and TLC overlay bioassay. All these techniques were used. I'll show you a small picture. Extracts were taken. After that, this different concentrations were studied. After that, biosensors. The biosensor I have used here is Chromobacterium violaceum for my studies. And after inhibition, I have checked whether this activities, whether there is an activity of the sponge extracts on Vibrio. And the last one was identification of the secondary metabolites. Which metabolite? Actually, a metabolite was identified before itself. I've just checked whether it was the metabolite which did all these activities, and it was done by purification of extracts through HPLC and UPLC, and this structural analysis through NMR and GCMS. This is the HPLC purified compound. Later on, I'll show you, this is just the technique. And this was the sponge Oxinella donani, from which all this sponge extract, which was found to be more effective. Okay, now we'll go to the results. This is the first part of the result. Here, Vibrio identification was done. I have done 5 Vibrio species: Parahemolyticus, Alginolyticus, Harvey, and Cholera. All the 4 Parahemolyticus, Alginolyticus, Harvey, and Cholera. All these were isolated from shrimp, and Vibrio fishery was kept as a control. And this was the growth pattern of all the Vibrio species. I have assigned species as SPB1, SPB2, TC1, TC2, and TC3. And this is the growth pattern. And here we can see the biochemical characteristics of all the isolates. From this, I got into a small conclusion, all which were the bacteria I have used. And here all are the biochemical tests. And here I have the antibiotic sensitivity test of all the 5: Vibrio parahemolyticus, Vibrio alginolyticus, Harvey, cholera, and Vibrio fischeri. And here I have studied the antibiotic resistant status of the isolate. How this antibiotics responded to the isolates, that is the isolate which I have obtained from shrimp. In this, I have found chloramphenicol and furosemide as sensitive, and almost all others were resistant. Here the zone of inhibition of all the isolates I have studied. And here comes the agarose gel electrophoresis of Vibrio cholerae, Harvey, alginolyticus, fischeri, and parahemolyticus. And next comes the phylogenetic tree. I have done this just by using a software. I have done this to confirm whether the isolate organisms were the ones which I had in interest. Vibrio Harvey, and this is the phylogenetic tree of Vibrio alginolyticus. And here I have Vibrio cholerae, and the last one, Vibrio parahemolyticus. And this was the Vibrio fischeri was used as a control. And this TLC, it depicts the acyl homoserone lactones. Acyl homoserone lactones, which are the autoinducers. These autoinducers are used by the bacteria for their communication. And this TLC represents the acyl homoserone lactones isolated from the this bacteria, which they are using for communication. This is the TLC of isolates. And here I have the HPLC of all the isolates. Different acyl homoserone lactones are there. C4HSL, C6HSL, 3-oxocet. All these different types were found in was secreted by the bacteria, which they use for their communication. And this is the GCMS of hydroxyhexanoyl homoserone lactone. And next, I have the sponges. These were the 5 different sponges I have used: Sigmodosia species, Dendrilla nigra, Haliclona exigua, Calispongia species, and Oxinella donani. All these 5 sponges I have taken for my study. In that, I have used different solvents at first. I have used chloroform, butanol, ketone, methanol, and methanol dichloromethane. From all these solvents, it was found that methanol was more effective. In all these 5 species of sponges, methanol had a good extract. It extracted the compound more than the other. The other solvents. So chloroform, butanol, acetone, all were discarded, and methanol extracts were used. And this is the preliminary antibacterial efficacy I have done for this Vibrio species. From that, it was found that Oxinella donani has almost similar effect as oxytetracycline, which is the common antibacterial used for Vibrio. Almost Oxinella donani, it showed almost similar, maybe more. Some cases it showed, in some organisms it showed more activity, like to oxytetracycline. And these are the plates which was used for antibacterial activity. And here I have the MIC of highly active sponges. From the previous slides, seen that Dendrilla nigra and Oxinella donani, both these sponges had greater activity. So later on, only those 2 were selected for the activity for studies: Oxinella donani and Dendrilla nigra. And here
Yeah, now it is better.
Sorry, sir, I had to mute the person. Sorry.
Yeah, and then if you can make a full screen, will be a much
Ah.
useful. See the slides in better way. Try to do the full screen.
Okay, sir, I'm doing it. But
Don't worry.
yeah, it's okay.
Wonderful, very good.
Okay.
Fine.
Okay, and this is the growth pattern after administration of for Vibrio Harvey. This is the growth pattern, and here we can see this. The first graph shows Oxalia donani, the sponge, which I said was better for all the organisms. For for the elimination of all the organisms. And here we can see the growth pattern for Haliclona exigua. And next, this is the pattern for Calispongia diffusa. Here we can see for Dendrilla nigra. The last one for Carnosa. And this from all these 5 graphs, it was proved that Dendrilla nigra, this one, and Oxalia donani, both these sponges were more effective. And for the later on studies, only 2 of the sponges were used. And for all the organisms, it was the same result, almost the same result. For Vibrio cholerae also, we can see Oxalia donani and Dendrilla nigra had more effect. For Parahemolyticus also, the graph was same. There are negative values also in the graph. Actually, my guide, he said, whatever you get, it should be plotted. This is a control organism, fishery. Even fishery also showed inhibition in for Oxalia donani and Dendrilla nigra. Next, we can see what was a bioluminescence assay. For that, the same, all the 5 sponges we used, we can see Oxalia donani had inhibited organism. Bioluminescence is actually in the previous slides I have mentioned, bioluminescence was due to the quorum sensing producing with quorum inducers. So when this sponge extract was used, bioluminescence was reduced. We can see in Oxalia donani and Dendrilla nigra, considerably it was reduced for all the organisms. This is the pattern for Vibrio Harvey, and the second one is the pattern for Vibrio alginolyticus. Again, you can see the third one for Vibrio cholerae, Vibrio parahemolyticus, and Vibrio fishery. In all the studies, it was proved Dendrilla nigra and Oxalia donani had better effects. And this is the micro titer plate assay for different concentrations. Here also, the first one depicts for Oxalia donani and second one for Dendrilla nigra. From this, it is proved that Oxalia donani is more efficient than Dendrilla nigra. And the biosensor strain used here is Chromobacterium violaceum. And for this report, even for the reporter bioassay, it was proved this second plate is for Oxalia donani. It was proved Oxalia donani had more effect than other organisms, than other sponges. And this is a TLC overlay bioassay. All these proved Oxalia donani had more activity than all the others. And this is the HPLC spectra. Actually, this was already it was donazole. The compound was already identified. I just had to check whether it is this compound donazole. Yeah, this is a UPLC of donazole A. Donazole A, there are 2 compounds, donazole A and donazole B. Both these were identified before, and I just had to check whether this compound is responsible for this activity, for the elimination activity. And it was proved that both these compounds were effective. And this is even through NMR spectra, it was proved donazole A and B. This compound is effective. And this is the structure. The structure was already depicted by another 2 scientists. I just had to see it. And coming to the conclusion, the work concluded that marine sponge, it can effectively block bacterial communication. That is, it has anti-quorum sensing potential. So for that, various antimicrobial potentials were studied. They were used for arresting it. And after that, even after studying, it was found that Axilla donani, a potential sponge, it has enormous quorum sensing activity, and the fraction 3 of Axilla donani, which was a methanol extract, is responsible for this activity. Thank you. And that will be the end of my presentation.
Thank you so much, ma'am. Thanks a lot for your most informative and valuable lecture. Now, ma'am, there are some questions in our chat box. Can I ask?
Yeah, sure.
So, Dr. Prakash, he is asking that how anti-quorum sensing agents was identified.
Now, all these marine sponges, in previous studies, it was all these marine sponges had activity for cytotoxic activity. They had antibacterial activity. All those were there. Even our symbol garlic, it also has antibacterial activity. So my area was just to look for if it is this antibacterial activity attributed to anti-quorum sensing activity. So from all this, it was proved that it's anti-quorum sensing activity, but only if we go into further specific studies, we can completely prove anti-quorum sensing. This sponge has anti-quorum sensing activity.
Okay.
Now that is what type of assay exactly you perform to characterize that sponges as the anti-quorum sensing inhibitor. It is first is the well assay only, right? Antibacterial activity assay. But in antibacterial activity assay, we see a clear zone of inhibition. In quorum sensing inhibition, we see a diffuse zone.
Yeah.
Did you do that assay or you had some other control? And then other thing was that
I didn't go for that. Sorry, sir.
Exactly. No, you use that indicator bacteria, right? Chromobacterium. What was its role? Yes.
Chromobacterium, it produces color from the so that was used as an control. Control means to check whether this quorum sensing because I didn't go for animal studies or something like that. So for that, instead I used Chromobacterium violaceum since it had it produced color. When just checked whether this sponge extract was added, whether Chromobacterium violaceum was inhibited or not. From that only I reached to the conclusion that it has this inhibition activity.
And then that final substance which you identified was from the purified one? Is it a purified one?
No, sir. All this donazole, it was already identified.
Yeah, but then
it was
how
already previously it was already identified. I just checked whether it is this compound. For that,
you did not isolate from your sponges.
No.
How you concluded that this dolazole is the responsible thing?
Donazole, it was already identified. Donazole had antibacterial activity.
Yes. Now, what is the relevance with reference to your study?
Sorry,
what
sir?
way dolazole is related to your study?
Yeah, me and my friend, both of us did this topic. She also did animal studies and all. Okay. So I just did the microbiology studies and she did the animal studies. Even she led to the in that she actually identified this organism respond to. She actually identified 2 compounds and one compound characterized. The second compound was characterized. Already it was characterized by some other scientists. She also could characterize this and she did animal studies and in that it was proved that this compound, while using this compound, diseases were reduced. So I just included it in my...
Yes.
Ma'am, my question is, other than Chromobacterium violaceum, which other indicator organism can be used for anti-HL activity?
And we can, since we are not going to details of it, we can use all, I think we can use all the color strains. So it will be visible,
Okay.
which produce organisms, yeah, which produce colors. Maybe I think Serratia or something like that.
Even micrococcus also can be used. Micrococcus.
Yeah, yeah, Micrococcus, Serratia,
All
all those produce color,
Serratia
yeah,
actually
pigments.
produces very good pigments. Exactly.
Yeah, all those will produce pigments, so it will be easier for identification, I guess.
Okay, ma'am. Thank you.
Otherwise, we will have to, since my friend also did the same topic on animal study on animals, from that only we identify that disease were reduced. Otherwise, we had to do after characterizing all these compounds, we had to go for animal studies. And I think the next part, maybe we can look for, after characterization of the compounds, we can look for the toxicity, and then medicines or something can be done. I didn't go for all those. I just concluded in this way.
Thank you, ma'am. It was nice presentation.
Thank you.
Any other questions? Okay. Thanks a lot, ma'am, for sharing your valuable knowledge with us. It's really worth for all of us. Thanks a lot, ma'am.
Thank you, ma'am.
Yes. Now, I heartily thanks to I heartily thanks to Dr. A.M. Deshmukh sir and Microbiology Society of India for organizing such an valuable, informative, and knowledge-boosting lectures for all of us. I heartily thanks for this. Then I thanks to all the members who have joined this meeting. And I again thanks to all the organizing committee and Microbiology Society of India for giving me opportunity to anchor this session. It's really a proudful thing for me. So thanks a lot for all of us. Thank you so much.
Thank you, Amaja ma'am, Nilima ma'am, Prakash sir, Kavita ma'am, and not but the least, Arvind Deshmukh sir for inviting me to this lecture. Actually, I couldn't complete my studies. I just had to wind up soon. So if someone is interested, maybe they can continue with the topic and identify all this. Even toxicity was also part of my study, but unfortunately I couldn't go into it. Maybe someone else can go into it, and we can reduce the use of antimicrobials. And with that, I would like to conclude. Thank you.
Thank you, ma'am.
Thank you, Dr. Noah, and good luck.
Thank you, sir. Thank you so much.
All the best.
Thank you, sir.